My name is John Charles. I am delighted to be the introducer, not the moderator, but the introducer for this panel on the topic of the twin studies and inevitably the connection with the omics, the various kinds of genetic and other information that can be acquired from studies like the twin study. This is a component of the one-year mission that we just spent an entire panel talking about, and I will tell you that when this idea was first proposed, I was against it. I know this is a surprise.
There's a pattern.
It's a pattern developing. And I will tell you the story behind that is when Scott Kelly was designated as the crew member, Julie Robinson and I were briefing him on his experiments because he was going to go the next day, I think, and talk to the press, and he wanted to have some information on what he would be doing on his mission. And as we got up to leave, he said, well, I got a twin brother. Do you think the press is going to ask me anything about that? Are we going to do anything twins related? And I jumped ahead and said, no, that's a stunt. We would never do that. You've flown 4 times with your brother. Your brother's flown 4 times. We've never done anything about it. Don't worry about it. I came back and talked to my boss, Dr. Barrett, and said, think about this is the question. Can you imagine the press asking that question? And Dr. Barrett said something like, not so fast, John. There might be something here. And now, of course, I'm a believer in the value of the twin study because it does provide us insights that we do not yet have through traditional techniques. Those are the techniques that I'm trained in, for greater understanding of the changes that happen in the human body in spaceflight, delivered to us on a platter by the purely random chance that we have a flying twin and a ground-based twin on this one-year mission. So it's a beautiful tie-in of new potential insights from the one-year mission for the benefit of preparations for going to Mars. And it's a wonderful introduction of 21st century techniques into at least one 20th century cranium about how we can solve the problems we have more efficiently and more expeditiously in the future. So with this, I would like to turn the panel over now to its moderator, Dr. Barrett, who will tell you about the panel and what it's about.
All right, thanks, John. You're probably getting the idea that I don't really moderate anything. I kind of maximize everything, but for better or worse. So the twin study, again, when John first brought that to me, I will admit that was kind of my reaction too. It's a bit of a stunt, but you really had to think about it because we have some powerful new tools now that can turn this into a less of a stunt and more of a pilot study because of the precision of genomics, proteomics, all things omics, if you will, pan-omics. And the twin study was like the one-year flight in that we understand we're not going to get a big data set out of this. But aside from the fact that we can do one-on-one comparison, this became a forcing function to bring something to the spaceflight program that we had been trying for over 10 years to do unsuccessfully. And this was to bring the power of genetic studies in. There's a lot of trepidation about that, which I'm going to ask our panel members to address. But this really became a door that we really wanted to run through. So number one, we saw it as a good opportunity to bring this to the program. Number two, you can't not do it. When you have 2 crew members, both with spaceflight experience, identical twins, you will kick yourself forever if you don't seize this opportunity, at least get what you can out of it. So it really made sense. It definitely blossomed much further than we expected it to, and that's hopefully what we'll get out today. But our full hope is that this starts us developing what I'll just call the expressome of weightlessness. All of the gene expression, the products, the transcription products, protein products, everything end to end that goes with our current physiologic understanding of adaptation, which is pretty good, but not complete. But it'll never be complete without the omics understanding of it. So I'm going to, because we don't have much time and because I want to keep you guys awake, we're going to be a little rapid fire. I'm going to quickly introduce everybody, have them say brief comments, and then we'll open it for questions, I think, fairly quickly. So Dr. Serena Anand, physician, astronaut, aerospace medicine, internal medicine, also serves as my surrogate conscious at times. Dr. Ludmila Burakova, a world-class micro and cell biologist from the Institute of Biomedical Problems, was involved in the first study that we actually published on proteome products, urine proteome for cosmonauts flying. So that was a real groundbreaking study, actually, we published in the Blue Journal. Dr. Craig Kundrat, Deputy Chief Scientist for the Human Research Program, soon to be a headquarters, I won't use the word bureaucrat again, but facilitator, a very insightful guy who pretty much has a grid map of the twin studies for a nervous system here because of the work that we made him do. Dr. Christian Otto is our Chief Scientist for the Vision intracranial pressure risk, if you will. And he knows that field cold, and I'll have him talk a little bit about the opportunity the twin study presented with the VIP issue, and just to talk about the VIP issue in the context of the 12-month flight as well. Yuri Voronkov from the Institute of Biomedical Problems, one of the absolute international specialists on standards for spaceflight, and I might ask him to address the implications of the genetic studies for future selection of cosmonauts and astronauts for spaceflight, or at least how we might fly more informed if we don't use them for screening tests necessarily, but how we might optimize countermeasures or other means of prophylaxis. So with that, I'm going to ask Dr. Anand to address some of the ethical considerations of handling genetic data on astronauts and cosmonauts. As you know, we're kind of a small population of people. So.
All right, good afternoon. Thanks again for the introduction. And again, we know we're sitting in between you and food and refreshing beverages. So just to give people a little bit of background, I know a lot of the folks in this room are familiar with the data protection policies we have in place currently at NASA. And a lot of us in this room also sit on boards such as the IRB or the LSAH, which is our Lifetime Surveillance of Astronaut Health Advisory Board. Our group is charged with making sure that our astronaut medical and research data remains safe. We do review several studies. When investigators ask for attributable data, we try and help them get the best science that they can. We also look at things such as who on the PI team is trained in data protection or city training. We look at where that data is stored. And even if data gets to publication or is presented at a conference, we will take a look at that data to make sure if there happens to be any attributable information in there that either the individual astronaut is aware and consents, or that it's kind of scrubbed from the presentation altogether. So that is your kind of, I'll call it your nominal medical and research data. When you turn to genetic data, and this goes without being said, it now becomes very personal. Dr. Barrett and myself, we already have several astronauts in the office when studies come forward will come up to us before signing a consent form to say, is this study important? What science gap are we filling? Do you think it's safe for my data to go forward in this fashion? I've had that happen several times because sometimes they're just not sure and they want to make sure, and we like to think that they trust us in that matter and they know we sit on these boards that protect this data. So when the issue of genetic data came along, the questions of, how do we begin to protect genetic data? What happens, or what sort of secure server are we going to store that genetic data on? What happens if that data gets released accidentally? So what crew members will ask us is, how does that affect me for future flight assignment? Are they going to use that for selection standards, for retention standards? Could this genetic data possibly affect my progeny, my children down the line? These are the very personal issues that confront them and what we get asked about. So because of that, NASA really had to begin to shift gears. So for many years, we've been looking at medical and research data, but when you throw genetics in the mix, it becomes a lot more complicated and a lot hairier. So one of the first things we did was to pull down, and we had a big genetic summit not that long ago here at the agency, where we pulled down many governmental agencies, lawyers, folks who deal with genetic studies, certainly more frequently than we do. So we could meet as a group and say, how do you guys protect your data? Should we consider astronauts a vulnerable population? And this is a question that came up during the summit that we had, and we never really came to an answer, because the topic of a vulnerable subject or a vulnerable population usually applies to children, pregnant women, or a population that could be coerced. So these are some of the questions that we dealt with, and they weren't easy to answer. Even the agencies such as the National Institutes of Health, looked at us and said, wow, you've got a very unique study population here, very low numbers. We understand that if some data leaks out about one person, it could affect many more than just that one. And so we all began to take a look at it over a couple of days and ask questions such as, how should we provide data to PIs? What should we do with that genetic data once the study is complete? Do we destroy it? What sort of server should it be stored on after that? How do we even begin to broach publishing in journals or presenting at conferences? And so what the agencies asked us was, well, tell us how you protect your data now. And we went through all the safeguards that are already in place, that NASA puts into place. And what I found really reassuring was that they looked at us and said, you guys are doing all the right things. And I looked at all the boxes that the NIH had to check when dealing with very special populations and going through their IRBs and looking at genetic data. We filled every one of those. So we are on the right track. It left me with a sense of great confidence that, sometimes I fear people feel that we're too conservative with protecting data. But I look at what the rest of this country does when it protects genetic data, and I think we're doing the right thing. So what we tell our office, and I think folks in our office know, is that the science is coming. It's a big train, it is coming, but we're going to be prepared to handle it. And what's important for us as kind of stewards of that data is to remember how responsible we are for it. And just to protect the health of the astronaut, protect their data on down the line. And by virtue of that, if they know those protections are already in place, then even though our numbers tend to be low for a lot of these studies, folks will feel confident in signing up for them. Thanks.
Okay, appreciate it. Thanks, Serena. I'm sure we're going to come back to that in some questions. And Dr. Burikova, if you could give us an idea of the range of omics testing that might be relevant to human spaceflight.
Before starting talking about omics, this is proteomics and metaomics. I would like to support Serena here and say that definitely genetic information has to be handled especially carefully, not to make sure it is not leaking, not to make sure it is kept confidential, but also in how this information is presented to cosmonauts and astronauts, because scientists, for example, found a modified gene. Does it make sense to talk about it? Because a structural gene is changed structurally, but it is functional, and cosmonauts' relatives will think that they may be defected. No. This is something that we have to approach in a special way. As to other omics, for example, such directions as proteomics and metabolomics, here there may be difficulties of a different plane. We have huge databases, and special informational processes are necessary to process this data and understanding of the data. For example, what is proteomics? If to think about it visually, you can take a typed page, cut it up, mix it up, and look at every detail, at every piece of it. Yes, we probably will recognize some letters, but we will never be able to put the original text together. That is why for any research in omics, proteomics, metabolomics, we need to have some monitoring, controlling experiments that will let us get rid of the noise and will allow us to explain, to see metabological and other systems clearly. When other institutes started studying proteomics of urine after spaceflight, prior to that, we had for many years run research of urine for healthy people, subjects that live in normal environment of different age, people who have different professions, to make sure that we will not have the noise information, so that we will really understand the protein profile changes during flight. That was the first issue. The second problem was that since the research is looking into the expression of genes or change of metabolic reactions, we have to have very well and very good understanding of correlation or physiological type. For example, expressing gene and biochemist expression of the genes may be changing, but it does not mean that proteins are changing. The expression of protein may not be detected over there. That is why sometimes we should not be running for sensations. We have to continue that link, genome, proteobiome, and physium. That is the physiological condition of a human being that we are observing as monitors during the flight or after flight. The research twin studies that we are trying to describe, this is a unique capability for manned flight that is definitely unique, and that is something that was given to us by nature, and it is a great chance that the twins, the same brothers, were also trained astronauts. On the other hand, this twin study, this is all studies in biological satellites. For example, one group is in space, the other group is of mice staying on the ground. And we can also run similar studies between the two groups and compare in 2 or 3 months what the results are. That is unique for life. This is variation ability for linear animals. It is giving us a possibility to get adapted to different environment. This is what is known by chemists, epigenomic differences. When genome is one and expression is one, but the genetics is providing us with variations that allows the organism react differently. Whether it is good or bad, we will answer these questions later. That will allow them to adapt.
And the fact that omics will continue going forward, we have absolutely no doubts in that. Right now, we have huge advantage of very fast development of all the tools and equipment used for genomic studies. But of course, we need to keep an eye on data processing and keep an eye on all the random data that we receive as well. I can't say that the future is behind the omics. However, they are an important part of our future diagnostics and studies.
Thank you very much. This is really interesting, and I could ask a lot of questions. Let me ask one quick one. Is that how difficult are these techniques? Are they widely available? If you do your tests in Russia, are they 100% comparable to our tests in the U.S.?
Ну, на самом деле, если вы имеете в виду, насколько трудно.
If what you mean is how difficult it is to compare the two, for example, proteomics, when we duplicate the experiments, and we're talking about microliters worth of samples, it's difficult to separate the noise, but it is possible, and it is possible to identify the important markers that we're studying.
I would like you to give a 3-minute and 14-second overview of the of the 10 studies that were selected for the twin studies, if you can, and you don't have to hit all 10, but no.
Okay, so. The intent was partially, well, the intent was to span the molecular up to the physiological and cognitive realm. So very briefly, in like a sentence per investigation, there is one that's looking at telomere length. Most of these have to do with peripheral blood mononuclear cells. Telomere length and telomerase activity as a measure of stress and aging. behind all these omics questions is how will Scott in spaceflight, how will his time course differ from Mark Kelly on the ground? Mark is not going to eat space food for 1 year. He is not going to exercise according to Scott's exercise prescription. He's not going to sleep when Scott sleeps. He will be what we call free range. He will be allowed to do whatever he wants. So that's the opposite of your normal scientific experiment, but if you think about it, this will give us a very good idea about normal day-to-day, month-to-month variation over the course of the year. So if we see changes in Scott that are significant relative to Mark, there's a good chance, well, it's tantalizing. We need to follow that up with conventional studies. So that's a kind of an answer to the n equals 1 or 2 question. It'll be a clue, but we've got a nice level of variation. So telomere length and telomerase activity. Epigenetics, the methylation of DNA and the modification of the histone proteins that pack DNA, that's being looked at. Those are involved in regulating gene expression. We're looking at RNA transcripts, what are being produced in these cells, how are they being modified by methylation and other processes. Continuing through the central dogma, we're looking at proteomics, doing both targeted proteomics of specific proteins, but also using mass spec to look at a broad panel. Same story with metabolites, looking at targeted metabolites with sensitive assays, but also with mass spec looking at broad panels. At the cellular level, we're looking at the immune system, the B cell, T cell repertoire, cytokines, et cetera. Also at the cellular level, we're looking at the microbiota in the gut. So looking at the microbiome and seeing how Scott's life up on station, eating space food in a relatively sterile, clean environment, how his gut microbes might vary over time compared to Mark. And then we go from the molecular and cellular level up to the physiological, picking up on investigations that John Charles mentioned earlier that were already occurring on the space station. So these pick up Mark Kelly as a ground subject. So this is looking at cardiovascular function, fluid shifts, and then also cognition. So Scott would have been doing cognition also, but we pick up Mark as a ground subject. And then the 10th investigation is very important. It's Mike Snyder at Stanford University. He's going to integrate all of these studies and all of this data. And the reason is, and this was part of the inspiration behind the call for the twin study, is in 2012, same year that Scott Kelly and Mikhail Kornienko were named to the twin study, he published a paper in Cell with an N of 1, that was himself actually, where he did a 14-month study of himself doing the genetics, the DNA studies, RNA, proteomics, metabolomics. And just to give you one example from that paper, he has children at home, and he had 2 viral infections during this 14-month period. And when you look at the transcript in these peripheral blood mononuclear cells, there was coordinated up and down regulation of expression in several different pathways, some of which were anticipated in advance, but others were not. And the same up and down regulation happened with both infections. So it's only a N of 1 subject, but that temporal history really is a strong indicator that some of these pathways that we did not know about previously or know about their role in viral responses can be significant, even with an N of 1 study. So that's the portfolio, basically spanning DNA, going through the central dogma at the molecular level, then up to physiology and cognition.
Brilliant. Okay. So, yeah, I think that underscores the fact that it's the new tools that we have now that make this more than a stunt, quite frankly.
And just an extra correct question to carry on your last question. Within the twins team, what they would say is when you ask a question about a specific assay for a lot of this omics, it's developing so rapidly that if you ask the question 6 months later in terms of what technique to use and how to do things, it will differ. So one thing that is going to be done with a lot of Scott and Mark's samples is in the fall of 2016, 6 months after return, when the last big sample take is done, then there will be there'll be a lot of samples in the freezer that will then be run on all the machines together at the same time, so to minimize batch effects, because there could be technology's changing and a lot of these assays are sensitive. So that's one. Things will be relatively quiet in the twin study from the molecular realm until fall of 2016, next fall. Then it'll be an explosion of data.
Okay, perfect. Thank you. Okay, and Dr. Otto, I'd like you to give just a few minutes overview of where we are on the vision intracranial pressure syndrome, I'll call it that, and how the twin study, having a control on the ground, might map to further understanding. But in particular, we needed to put VIP somewhere. The fact that we're flying for 12 months, a year, with this risk that we know is correlated with time is kind of a big deal. So I'd like you to address that as well.
Yeah, thanks a lot, Mike. And so we're really excited about the participation of both the one-year crew members that you've seen today, and they're actually involved in two of our flagship VIP research studies in flight, both the ocular health study and the fluid shift study, which you heard about earlier today. So what is the VIP risk? If we look at the number of crew members who've flown on station who've been adequately categorized, there's 33. So of those, 22 have some kind of eye structural change or change in function. That's roughly 70%. And 7 of those 22, or 20%, have been affected to a greater degree, showing optic disc edema of at least Friesen grade 1, most of them, and there's one grade 3. So we hypothesize that the microgravity fluid shift increases the resistance to cerebral venous outflow, and this increases the intracranial pressure. So do we have any evidence for this? There's some evidence. We know that in-flight cardiac preload increases. We've seen some evidence that the internal jugular vein diameter also increases. We have 5 of 6 crew members have had an elevated CSF pressure post-flight. We do not have a pre-flight. That study has just been funded, actually. And then we have MRIs that have changes consistent with intracranial hypertension. So the ocular health study has been running since 2012 now. We have 6 subjects who've completed the flight phase. We have 1 that actually has disc edema. And so this study is going to help us understand the susceptibility and who's at risk, post-flight recovery profile, which is really important because you've probably heard that there are some permanent changes in pathophysiology. We're trying to characterize the changes in the eye and the retina, changes in brain compliance and blood flow, pre- and post-flight, and changes in brain structure. An interesting piece of data I can share with you now is that we're actually seeing changes, retinal changes that are persisting post-flight, even in the non-cases, even though they're not considered cases, up to 180 days. So we're still, this data is coming in as we follow them to 1 year post-flight. So that's very interesting. In terms of the fluid shifts experiment, this is a very exciting study because unlike ocular health, which is an observational study, this is an interventional study. So the Chibis ML lower body negative pressure suit is actually being applied to give us a sense for how the physiology is changing when you're measuring eye, brain, and cardiac function both before the suit is on, when the suit is on, and when the suit is off. So as you know, the suit provides a 1G-like profile of body fluids using vacuum. And so we can look at these interventions and whether cephalad fluid shift is causative as we think it is. One final key aspect of the fluid shifts experiment is that it's actually measuring compartmental fluid shift using deuterium labeled water. So we'll get a very accurate assessment of the fluid shift. So 2 points to finish on why the 1 year studies are of particular importance. One is the concern for the existence of a dose response. And I think there's some retrospective data that does suggest there is a dose response when you look at the 3 last shuttle flights and then you look at what we're seeing from the 6-month ISS flights. The severity, or they're revealing that the severity of changes and the frequency of changes are much higher for the 6-month flights versus the 1 to 2 week flights. And actually, Dr. Larry Kramer is here. He published a paper on structural changes in MRI and definitely found changes between those 2 groups. Finally, the twin study is exciting and of great importance because we're going to be able to compare Scott Kelly, as the other panelists have suggested, to his twin on the ground, and we'll probably have characterized his VIP status really well. So we'll be looking at those old MRIs of his brother. Thanks.
All right, thank you, Christian. That was a really good summary. I would ask more questions except for sake of time, so I want to move to Dr. Voronkov. And Yuri, I should have asked you the obvious question first, which is what selection criteria did you use that might have been different in selecting the 1 year crew member as opposed to a normal 6-month crew member? And if we have time after that to talk a little bit about how the genetic studies might inform some of your new selection criteria.
Thank you, Mike. I wanted to say actually a few words about something completely different, but since you are our lead and since you are present at our meetings sometimes, you know me well and you directed me in the more productive direction. So I know that Dubinin, Parfenov, and many other famous scientists worked in the area of genetics back in the 60s. And initially we worked with plants and animals, and then we did human studies, including twin studies. And we thought that we need to narrow down the scope even further and study myozygotic twins. But since you already outlined what I should talk about, I can tell you that there is publishing of environment and molecular ontogenesis. This is a published article that was published in 1997. So all that information that I wanted to talk about is available in that publication. Molecular from experienced Soviet cosmonauts. I think that's another important article, another study, which is a good foundation for our work, and that also directs us towards unified experiments, unified research, especially in the area of omics. We need to come up with a joint approach so that we can move forward together. As far as the selection process goes for astronauts and cosmonauts, at one of the most recent meetings at Montreal, we talked at length about this very difficult issue. And Serena mentioned we have a lot of advancements in science and technology, but we have to be very careful with handling astronauts and cosmonaut study results. But I know that there is a certain aspect of nosology, certain studies and data that points to predisposition of breast cancer or prostate cancer that needs to be pointed out, and maybe
okay, great. That was a bit of a whirlwind, but really, really informative. I think I'd like to take about 5 minutes or so for questions if you have them, and then try to let people out. General.
Dr. Byrne, I'm just an engineer, not a doctor. But you know, you talked about Doc Byrne, I'm just an engineer, not a doctor. But you talked about the intracranial pressure up here and all that. You know, just what about putting them on a centrifuge for a couple of days, just regular like an airplane, eyeballs down? That's going to decrease that pressure up here. The fluid's going to flow out. Maybe get them a G and a quarter.
Well, you're a very insightful engineer. I will tell you that at a time when I thought that we were retiring our risk for bone and muscle in zero gravity, the VIP syndrome arose, and it's renewed our interest in artificial gravity in general, to the point where we convened an international summit to talk about it. So it's a big contender, actually, for driving artificial gravity that we didn't have before. So that could be either the centrifuge or spinning the ship.
Oh, okay. I was thinking just recovery for the individuals. They still have some. That's going to reduce the pressure up there, just pulling it down.
Right. So I'll circumvent Dr. Otto by noting that what we see is actually permanent remodeling of the anatomy. So you'll be able to pull the fluids down with centrifugation, but most likely if you've flattened your globe or distended your optic nerve sheath, that's permanently with you. So it's just something we know empirically, not something we would have guessed. That's a good question. David, do we have a...
At the end of the twins, I guess this is for you, Craig. At the end of the twin studies, as there are new discoveries around metabolic and other biomarkers, which is an extremely high priority area for NIH and nearly every federal agency now, identify phenotypic vulnerability in any number of domains. We just had a big meeting at NIH on it. As those discoveries come about in other labs, will you have biological material available that could be checked, that could be used to see if Scott or Mark carried a vulnerability, a marker for a vulnerability in a given area? And what I'm talking about is using this stuff to start moving towards predictive biomarkers for who, and dare I say it, is fit for long-duration space flight and who is not. And the dark side that we never talk about is we are not all equal, not even close. And it doesn't matter how much we socially create our equalities, we are biologically different, and the phenotypic differences far outweigh what changes in our lives. We carry differential vulnerability to cancers and all sorts of other things. And so I think making sure that this biological material is available for biomarkers research later on, just to determine whether, aside from the fact that it's going to be looked at from gravity changes, just transition to microgravity, it may have some value later on to determine if either carried a biomarker that would indicate something that would be risky. We just published one in PNAS on a metabolic biomarker in rodents and humans for sleep debt. It appears when you're in sleep debt, and when you're not, it's gone.
Yeah, so the answer to your question, David, it's a very good one, is this is one of the things that came up in developing the genetic research policy at the agency. And the short answer is we will throw away nothing. The investigator labs, if they're all done and can't curate the material, NASA will take it in. NASA may unilaterally take it in, but the going forward with genetic studies, that's the default position, is that this is, as you kind of intimated, very rare material, and we want to save it for the advances in future and future analyses.
What about data sharing of it? In other words, because these guys get a staggering amount of information, if somebody wanted to look for a particular a pathway or particular analysis in lipids or something, will the data be available through some sort of a process, application to NASA to get it?
Yeah, the generic answer is yes. There too, we will be archiving all the data from the investigations. But when you get into the data sharing, as Serena kind of alluded to, you can how to actually do that, I think is something that will be determined in future, because, for example, giving wholesale genomic information is probably not the right thing. It's got to be targeted inquiries, and what's the system for vetting those inquiries? We haven't set that up yet.
Just one follow-up. You talked about the dark zone of who's fit to fly. We talk about that all the time, actually. Well, I'm not sure you've ever asked us, but no, that's okay. But we don't see the outcome as so binary as who's fit to fly and who's not. The outcome could be what countermeasures does this individual need, and the other outcome is that if we find that zero gravity really is only tolerable by X percentage, then zero gravity is not acceptable, and we need a technical solution for it. So again, as part of that fly informed, we have no reservations at all about examining the genetic differences between individuals.
Yeah, it's really about personalized medicine, et cetera. And we may find, you may find somebody's carrying a clear marker for vulnerability to one aspect of spaceflight, but not another. And then you have to decide, is it the radiation vulnerability that is a more concern? Which one can't we mitigate? And then, but I could see how this would play out down these channels of reasoning regarding what definition you're trying to hit for optimal crew.
Right, and you're absolutely right. I think part of NASA's charter, parts of which are written and parts of which are is opening up the space frontier so that others may follow. And so a wider population base implies good understanding and better understanding of how protective measures, which I see a thumbs up, so I guess that's good. Joe, Dr. Kerwin.
Yeah. Serena, I believe that NASA has preserved and banked biological specimens from previous programs. Does our current state of knowledge make those valuable in any way, and does it require additional protection?
I believe, and y'all can correct me if I'm not stating this right, but those specimens are protected under the same rules that we protect specimens from current astronauts, former, retired. And so I don't think there's any change in rule when looking at that. in fact, we do have a group when it comes to consenting. Let's say we had a PI that came forward and wanted attributable data from someone who flew during Skylab, for instance, then that still contact those people today to get their consent to participate in any sort of study. And we do protect the data very similarly.
Because there might be a correlation between a previous finding and a genetic sample.
Absolutely. And we also try and, also operate under the fact that, if we do release one astronaut's, attributable data, regardless of how long ago they flew, again, they don't operate in a bubble. That could affect the others that flew with them because our N is so small, especially when you're looking at station missions. Suddenly you've narrowed it down to, well, the one person left it could be was female, and it's very easy to figure out who that was. And so we're constantly trying to pay attention to that. But, we want the science to go forward, but we don't want to forget all the pitfalls and traps that we may fall into along the way.
Eric, you got the microphone.
I was wondering, with the genetic study with Mark and Scott Kelly, this is going back to what Jay Bucky was saying about the random effects. Is there also going to be a period of overlap where they're both on the ground and both having the simultaneous genetic analysis to try to separate which effects are from microgravity and the environment on the ISS versus what is their genetic expression similarity during 1 year where they're both on the ground free running?
The short answer is yes. The pre-flight data collections were first done for Scott and Mark in October and December, so close but not on top of each other. In January, they were a week apart. Upon return, right when Scott returns, when he has a big sample take where we have Mark scheduled to do the same. There's a caveat to these schedulings, by the way. Mark is a volunteer citizen as opposed to a NASA subject, in the usual way. But the plan is that Mark has agreed to in advance is we'll have a big sample collection right as Scott returns, and again 6 months later, and those two will line up. So we'll have samples from both Mark and Scott at the same time.
A question for Christianata. I know this question does not pertain to twin studies, but it does pertain to VEEP. It's something very important to all of us, especially in the area of countermeasures. We know that resistive exercises can lead to the increase in thoracic pressure and eventually intracranial pressure. Of course, that would be true in the case if the breathing is done incorrectly during resistive training. So are you studying this aspect at all, and what is the status of those studies?
Good question. So let me just start off, I think, with our hypothesis is that resistive exercise may exacerbate the condition but not precipitate it. So let me give you an example. If on the ground here your supine ICP is 10, and let's assume for argument's sake it's 20 or 25 in flight, if you were to go and then use the resistive exercise device, 1 hour a day, 5 times a week, for periods of time during that resistance exercise, it will increase your intracranial pressure above what your space station baseline is. So for some individuals, maybe that's enough over an ISS flight to tip them over from not having clinical grade edema to having clinical grade edema. So actually, we do have a study that's ongoing right now. I've actually seen some of the preliminary results that I'm probably shouldn't talk about, but which compared continuous exercise, interval exercise, and the resistive exercise in a head-down tilt environment, and actually was looking at surrogates for the translaminar pressure gradient, the pressures across the optic nerve head, which we feel are very important. And there were changes with resistive exercise. It's interesting, though, that it doesn't always occur the way you might expect. So it's most likely is contributory. It's probably not the causative factor, because many of the symptoms that I mentioned have been documented in flight, the elevated cardiac output, the jugular venous distension, when resistive exercise wasn't happening.
This might be more of a comment than a question, but firstly, there are two other active astronauts, Craig knows this, that have twins. Their twins are not astronauts, which actually could be an advantage. They've never been in space, so they've never been subjected to space radiation. So potentially there is 3 sets of twins actually that NASA could, if they chose to. Again, obviously the siblings could not be compelled, and astronauts can't be compelled obviously either to participate. But there would have to be, a real element of volunteerism here. So anyway, there are potentially 3 sets of twins. The other comment I wanted to make is we live in the era of personal genomes. Jim Watson was the first person to have his genome sequenced and put out for everyone to look at. But Richard Gibbs was here actually last year at the meeting, and some of you will remember what he said. That he said something along the lines of, some of you in the room might be thinking of sequencing astronauts, and I don't know that that's that great of an idea, because if you come back 20 years from now, all of the astronauts will have had their genomes sequenced when they come into NASA. And I think that's something we should keep in mind, that this is like an MRI. This is like something that's just going to become very commonplace, that probably all of us in this room are going to have our genomes sequenced at some point, most likely. And so the last point to make is that's sort of tied to both those previous 2 points, is it's quite valid science actually, and I think Craig might have mentioned this, to look at N of 1s in genetic studies, particularly if you're doing integrated omics. If you're looking at DNA, RNA, protein level, metabolites, the kind of things we're doing with the Kelly twins, that's extremely powerful. And that's what Mike Snyder did and published in Cell. And the twin study is probably one of the most powerful, if not the most powerful study that's really ever been done. So NASA's really, in a way, blazing a real trail here. So anyway,
and
just some points to think about.
Just to follow on with that, with Jay Bucky's question about the nosebleed. That's a single scalar variable. Do I have a nosebleed or not? Compare that with the Snyder 14-month study. He had 3 billion data points. So correlation is not causation, but when you have 3 billion data points and you see coordinated things happening,
Yeah, no, I think the point with that was just random events. For this, I mean, if you're making multiple measurements, you establish a good baseline, you can still have significant results from an individual. It's just a question of generalizability, and that's just the only point I was making was, with a 1-year mission, you don't want to generalize it if it goes well, that all 1-year missions go well. By the same token, if it goes poorly, it doesn't mean that all 1-year missions will go poorly. But you can still get statistically significant results from the people who were studied if you design the studies right.
Okay, great comments. I think we maybe limit it to 2 last, and then we'll let you guys be free, and everybody here will hang around a little bit, won't you? And you can ask questions. Okay, Dr.
So can I say something slightly contentious maybe about genomes and genetic testing? These are very powerful techniques, but I find it surprising really that we're moving towards trying to select increasingly superlative individuals to prosecute these programs of exploration, when the history of exploration largely is to engineer that out of the system, just to engineer systems in which a wider section of society are able to engage in the exploration rather than a narrower set. And so I just wondered your thoughts on that really. I mean, this is an engineering organization, isn't it?
Go ahead and start.
I was going to say, I think one of the bigger challenges is first learning how to apply the science. So, you may be able to map an individual's genome, but part of my training is in preventive medicine as well as internal medicine. And what we're taught in preventive medicine is that genetics loads the gun and environment often pulls the trigger. And so trying to figure out how to take a very specific piece of genetic information and apply that to the outcome you may see or the performance level you may see is difficult. And so part of this is for us to learn. I think you're right. NASA it's pretty trailblazing at this point. I don't think we're going to turn around in a year and say we've got these great selection standards or retention standards we're going to apply. Now there are some genetic data points out there like BRCA or things like that we have seen a lot of data on, and we are able to maybe apply towards better screening for some of our astronauts to help prevent bad things from happening. How fast this moves, again, I called it a train before. It is coming. The science is coming. again, I think as long as we take that science and apply it as productively as we can, I still tell a lot of my students and residents, if you order a test, you make sure and tell me what you're going to do with the results of that test before you order. Don't just order it to get information, and tell me and not tell me what you're going to do with it. And so I think that's going to be the greater challenge in the next 5 to 10 years.
Craig, did you have a comment?
No, I think I agree, and I would just also say it's kind of an amplification of one of Serena's points. It's early days. We're just, there's a couple aspects to this. We don't know how powerful the predictive tools will be, and so how we actually implement those will probably just depend specifically on where the powerful predictive power is. Second, one thing that we've faced in a concrete way with Scott and Mark Kelly is the odds of them having a really notable surprise in their genome, at least for this point in their life, is very low. I mean, we're already selecting phenotypically, a set of individuals who are likely to be free of a whole set of pathologies. So, there are other, later in life, issues that one might discover. But for the mission, it could well be that the phenotypic screening that effectively is being done right now is close to good enough, or won't be affected that much by the genomics. That's a possibility.
And I would add that, again, we tried to bring genetics into the program about 10 years ago. Two things stopped it. One of them was data privacy. The other was exactly what you said. Would this be applied to selection at a time when we really don't understand how that selection could be, practically, what its practical meaning is? And we really aren't out to choose superlative beings. You'd be a far cry from that for certain now in the astronaut office. But genomics is supposed to make everybody's world better. Personalized medicine, better understanding of physiology, and that should apply in spaceflight as well. So we have to apply that carefully. But again, we want to fly informed. We don't want to fly super beings. And weightlessness, if you really look at a long run over decades, we don't want to be in weightlessness for long periods of time. So we're waiting for other technologies that can fly us faster or provide artificial gravity. But this is an interim period, the way I see it. Nick, I think, take us home.
This is a very interesting panel, and I think it's stimulating me in some way where there's some kind of a methylation occurring and an out-of-the-box gene got activated. Here's the story. Here's the story. This is really very interesting. I'm sitting here thinking, apropos what Jay said when I said earlier, wouldn't it be great to have a bunch of twins and to study a series? And I'm thinking, boy, how many of them are going to be astronauts? Has there been any thought given by NASA to recruiting subjects, maybe through the private sector or using the private sector to recruit a pool of twins that might go up suborbitally, that might be studied? Is this at all allowable? Because that would be one way of maybe looking in a follow-up at other twins and doing other studies that might further and advance some of the genomic issues you're discussing.
Craig, you want to hack at that first?
I'm afraid, Nick, my only response is I'm so glad I can say that's so far above my pay grade. It's an intriguing idea, but a problem with the suborbital aspect is it's going to be such a brief exposure. Certainly, you can recruit twins for a study, but selecting them in for a population, it seems like it would be an awfully limiting constraint for building up the astronaut corps in future. So. I wouldn't hold my breath for it. But I mean, it's a tantalizing, idea, but I think it's a stretch.
Yep. Do you have a twin, Nick? Okay. Okay. Well, listen, we're way beyond what we should have been. So I want to thank you guys for hanging in there. I want to thank the panel members for answering very candidly and very informatively. I want to thank our interpreters. Spasibo za pomoc. You guys have been really, really good in hanging in there. And again, if there's any questions you want to ask privately or we're too embarrassed to air in front of everybody, come on up. So, thanks. Good questions.